My name's Gemma Klukas and I'm a research associate here at the Cornell Lab of Ornithology. I've been monitoring seabird diets using fecal DNA metabarcoding for a number of years now. So in this video, I'm going to take you through all of the steps that I use in my work in case you're interested in setting up your own seabird diet studies using fecal DNA samples. One of the major requirements for doing this work is that you can separate your pre-PCR and post-PCR lab spaces. So I have this lab here, which used to be our ancient DNA lab. It's now our fecal DNA lab, and I do all my pre-PCR steps here. And then further down the corridor behind me, we have another molecular lab, and that's where I do all my post-PCR work. And so being able to separate physically these two spaces is really important to preventing cross-contamination from your post-PCR products into your samples. Now, I'm very strict about the movement of people and items between these labs. So we won't do any work in the post-PCR lab before coming into the pre-PCR lab so that we don't risk moving any of those PCR products back into the lab where we're handling our raw samples, because that could be a big contamination problem if we were to do that. So this is our fecal DNA lab where we do all of our pre-PCR steps, so getting ready for the field, our DNA extractions and our PCR setup. We always wear lab coats in here and we're very strict about using this space and who can come in here. And the reason for this is that contamination can be your biggest enemy in a fecal DNA study. By the time the prey has gone through a seabird, that DNA is going to be highly degraded. It's going to be very low quality, low quantity, and so there's not much that we have to work with in our samples, meaning that it's very, very easy to accidentally contaminate your samples with DNA from your lab space, from other studies that are going on here. So we have to be very strict about keeping our lab space very clean so that our samples do not get contaminated by post-PCR products or anyone else's work that is going on in the building. Now, I would recommend you get all your tubes ready in the lab before you go into the field. So I use these screw cap tubes which have an O-ring in the lid because they can seal really well and they're not going to leak. To each tube, I then add my buffer, which is going to stabilize the DNA once we add the samples. So the buffer I use is DNA RNA Shield, which you can purchase from Zymo Research. And I really love this buffer because not only does it allow you to store your samples at room temperature, but it's compatible with DNA extraction methods. So you don't have to worry about removing the buffer from the sample once you get back from the field, like you would if your samples were stored in ethanol or RNA later. So I would highly recommend adding your DNA RNA shield to your screw cap tubes before you go to the field. I also add 2 different sizes of these zirconia beads to the tubes. Now these beads are actually used later in the DNA extraction, but adding them now is much easier while the tubes are still sterile, and it minimizes the number of times that we have to open the tubes later, therefore minimizing the chance that we accidentally cross-contaminate one sample with another. The last final step is to just label the tubes. I add a unique number to every tube so that in the field we can just record the tube number and then all other metadata can go onto a data sheet with that sample number. There are lots of different ways to collect samples. If you're in a colony of seabirds that are going to poop on you, like this single species colony of common terns, I like to cover my hat with a clean trash bag and just walk around and get pooped on. We can then collect samples straight off the plastic so they're nice and clean and fresh. Putting chicks in paper sandwich bags also works really well. We get nice clean samples from inside the bags, and we only need to collect the chunky parts of the poop, so avoiding the white uric acid. We don't need very much, just a pea-sized amount, and so that amount can go into the tube, and then you need to give it a good shake to mix the buffer with the poop. For recording metadata, we obviously want to know what species it came from, when and where, what substrate the sample came from, so was it from a paper bag, was it from plastic, Was it from clean clothing? Obviously not my clothes here, they're very dirty. And it's also a good idea to record the age of the chicks so that we can look for variation in diet with chick age. And there are lots of other ways to collect samples as well. You can put paper down around nests, or you can put birds into enclosures lined with paper so that we can collect nice clean samples from those surfaces. Now, it is also possible to collect samples directly from the ground, but we have to be very mindful that if we're doing this, it's going to be very easy to pick up contamination from the ground itself. So you just want to collect the top surface of the fecal sample in this case, and I'm really going to emphasize the importance of collecting field blanks when you're out in the field. So whether you're collecting from the ground, from plastic, from paper, you want to collect some negative controls from those surfaces to monitor for any contamination that you might be picking up in the background. So now we're back in the lab, and the first stage of the DNA extraction is to bead our samples. So those beads that we added to the tubes earlier are going to be shaken around inside the tubes, and that's going to break down any intact tissue and break down all the cells to release the DNA into solution. So I bead my samples for 2 minutes, but you may want to use longer. For example, if you're looking at the diets of insectivorous birds, because the tissues of those prey are going to be much harder to break down. Now, at this stage, we're going to treat our field blanks just like our samples, so they're included in this plate of samples here. I do my extractions in 96 well plates, but I don't put all the samples from one colony on one plate. Rather, I mix them up amongst plates in order to minimize batch effects. I also leave a number of gaps on my plate, so I don't fill all 96 positions. The first gap I leave is for a mock community. This is a positive control of known fish DNA that I use to always make sure my PCR and my bioinformatics are working properly. I then leave gaps for 2 PCR negatives and 6 further extraction negatives, and having these will allow me to differentiate from any contamination that might happen on the plate during the extraction or during the PCR setup. So once I've got all my samples laid out, including the field blanks, and I know where my PCR and extraction negatives are going to be, the next step is to centrifuge everything and then transfer just the supernatant, which is the clear liquid from the top of the sample, into a plate where the actual DNA extraction is going to happen. So our plate of samples then goes into our Opentrons robot, and this is going to run a modified version of the Quick DNA fecal soil extraction kit from Zymo Research. Now, you don't need a robot to do your extractions. Zymo makes some great kits that use tubes that you can do by hand, but the reason we use the robot is because it actually minimizes human error in these extractions when we're handling large numbers of samples at once. And so it takes about 3.5 hours to do the full extraction. But once we've finished, we're going to store our DNA in the freezer until we're ready to do our PCR. Now, primer choice is really important in your PCRs, and for identifying fish in the diets of seabirds, I mostly use these primers developed by Meyer et al., which are called MyFish primers, and they amplify a small section of the 12S gene, which is really good for identifying fish to either genus or species level. However, these primers are highly complementary to the penguin 12S gene, so for looking at the diets of penguins, I prefer to use mini-fish primers, which still amplify the 12S gene, but without amplifying penguin DNA. Then for looking at invertebrates in the diet, I tend to use these 18S primers developed by Julie McInnes in this paper, with the addition of a blocking primer which prevents amplification of the bird DNA. These 18S primers are great in that they're very universal and will amplify DNA from a wide variety of different eukaryotes, but they will also amplify the host bird DNA as well. So that blocking primer prevents amplification of that bird DNA and allows us to amplify a greater proportion of prey DNA. Now, I won't take you through the whole PCR setup because all those details can be found in papers, but I will note that I do my PCRs in duplicate. So from the one plate of DNA, I do 2 PCR setups. And with the MyFish primers, with the TrueSeq adapters that I've added, I've found it's really important to do the PCR setup as fast as possible and keep everything as cold as possible to prevent primer dimers from forming. We then run our duplicate PCRs in our main molecular lab, and these plates will never go back into our fecal DNA lab because the PCR will amplify the DNA to very high concentrations, and we don't want that to get back into our original samples. After the PCR has finished, we use a small amount of the PCR product to load onto a gel so that we can visualize how well each sample has amplified. And we load the replicates side by side, loading both plates at once so that we can visualize all of the samples. So after our gel has had some time to run, we're then able to see how well our different samples have amplified. And while this is not the most accurate method of quantifying that amplification, it is the cheapest, and so it makes financial sense given that we're trying to run large numbers of samples. And so based on the brightness of the bands on the gel, I then dilute the samples to try and normalize the concentrations of all the different samples on the plate before I send them off to sequencing. We can also check that all of our blanks are blank at this stage, although we also send them off to sequencing because we want to treat them just like normal samples. I send the diluted PCR products to the University of New Hampshire, where they add unique dual indexes to identify the samples and the rest of the adapter sequences through a second round of PCR. They then normalize again, pool the libraries, and then finally sequence our samples. Now I use 250 base pair paired end sequencing, meaning we have a forward and reverse read for each sample that we submitted or each blank. So you can see here we've got all of our blanks, our mock community, all of our samples, in this case northern gannets, and these are in FASTQ format, meaning each file contains a number of sequences and their assigned quality scores. And I analyse all the sequences within QIIME 2. Now, QIIME was originally developed for microbiome studies, but it's just as flexible for any sort of metabarcoding analysis. And I really like it because it includes a lot of different plugins that you can use. So one that we use often is CutAdapt, also Dada2, and you can run these individually, but running them inside QIIME is really straightforward, and so that's why I use QIIME for all of my analysis. Now I've put all of the code that I use to analyze my sequences up on GitHub, so if you want to use this as a framework for how to analyze your samples, please do go ahead. Hopefully it's straightforward and easy to follow. And the other reason I like working with QIIME is that it has some really nice visualization tools. And so we can just drag and drop our QIIME artifacts into the QIIME viewer and then immediately start visualizing the data. So here I have some samples from shags and gulls in the UK, and we can start to pick out which samples contain sandlance, which ones contain sprat, and so on and so forth. So it gives you a lot of flexibility in visualizing your data before having to get into a lot of coding. So that should have covered all of the main steps, right up from preparation before you go into the field to what to do with your data once you get it back from sequencing. If you have any questions, please don't hesitate to get in touch, or if you would like to send samples here to the lab for us to analyze, then again, please get in touch and we can talk about that. Thank you.